Assalamu alaikum, dear students. Welcome to the course Principle in Animal Life 2. This is lecture number 9. The topic is Law of Segregation. This topic has been delivered in 2 videos and this will be the first one. Discussing agenda for the topic will be Mendel used the scientific approach to identify 2 laws of inheritance. Mendel's experimental quantitative approach. Then we will discuss few terminologies related with Mendelian genetics, character, trait, true breeding, and finally we will discuss the law of segregation. Lecture outcome. After watching and listening this lecture, students will know about early years of Mendel's life and his experiments along with certain genetics related terminologies. Introduction. Brown, blue or gray eyes, black, brown or blonde hairs, these are some of the phenotypic traits. They are just a few examples of heritable variations that we may observe. What principles account for the transmission of such traits from parents to offsprings? Hereditary characters are those characters which are acquired by an offspring from its parents, and heredity is the transmission of characteristics from parents to offsprings. Variations are the differences in the phenotypic traits between the parents and their offsprings. So we resemble in certain phenotypic traits with our parents, and there are some differences in certain other phenotypic traits. So what laws, what rules, what mechanism is responsible for these similarities and differences, and why we look more similar to our parents as compared to other individuals in the population. Now, the explanation of heredity most widely in favor during the 1800s was the blending hypothesis. So before Mendel's work, the predominant and most widely acceptable theory about the hereditary characteristics and the transmission of traits from one generation to another was blending hypothesis or blended inheritance. Blended hypothesis is the idea that genetic material contributed by the two parents mixes just as blue and yellow paints blend to make green. So if you mix blue paint with yellow paint, the resulting color will be greener. So in a similar way, according to blending hypothesis, the characteristics of both the parents mix together and the offspring exhibit characteristics which is a combination of the characteristics of both parents. In yet another example, when a black cow and a white bull mates with each other, so according to this concept, the offspring will be black in color with white spots or white in color with black spots. So according to this definition of heredity, the offspring exhibit the characteristics of both parents. This hypothesis predicts that over many generations, a freely mating population will give rise to a uniform population of individuals, something we don't see. We don't see this phenomena in a natural population, but this theory, this concept predicts that no change, no variation occurs, and the offspring exhibits the phenotypic traits, the characteristics of both parents. So, with the passage of time in such a population, the individuals will tend to have similar traits, and there will be no observable variation. However, this is not the case in a natural population. A natural population do exhibit lot of variations. So it means there are flaws in this hypothesis. The blending hypothesis also failed to explain how traits can reappear after they have skipped a generation. If you go back to the Mendels work, I hope you know about his work to a certain extent. So he crossed a tall plant with a dwarf plant, and all the F1 individuals were tall. So if that crossed followed the principles of blended hypothesis, then the F1 offsprings would have been intermediate in size as that of the two parents, but the phenotype of the recessive parent was not shown in F1 generation, and this phenotype was skipped in F1, but it came to notice in F2 generation. So this is yet another problem which cannot be explained by blended hypothesis. Why one trait is camouflaged or hidden in one generation and then suddenly reappear in another generation. The blending hypothesis. An alternative to the blending model is a particulate hypothesis of inheritance, the gene idea. So, after the advent of Gregor Johann Mendel, after the experiments of Gregor Johann Mendel, after his work, the particulate hypothesis gained fame because it was the gene idea that the hereditary characters pass from parents to offsprings in discrete units, which Mendel called as factors and which we know nowadays as genes. So genes are the discrete units of heredity, and they are responsible for the transmission of traits, characters from one generation to another. Now, in the particulate hypothesis, in this model, parents pass on discrete heritable units called genes, and these genes, they does not mix up, but they retain their separate identities, they retain their individuality, although they will not be expressed in a particular offsprings, but the genes will be there. An organism's collection of genes is more like a deck of cards than a pail of paint. How? Like cards, genes can be shuffled. They can be passed on from one generation to another generation, but they retain their individuality. They cannot be mixed with one another and give rise to some other combination. So the genes, they retain their individuality, they retain their discrete nature. Now, from where the modern genetics begin, and uh, which findings give rise to the particulate inheritance? Modern genetics had its genesis, its beginning in an abbey garden where a monk named Gregor Mendel documented a particulate mechanism for inheritance using pea plants. So Mendel worked with pea plants. He studied different characters. He performed 37,000 crosses. He kept a very detailed record of all his crosses, and on the basis of these findings, he formulated the two laws of genetics, and that was the beginning of the modern genetics. Mendel developed his theory of inheritance several decades before chromosomes were observed. The chromosomes were observed in 1880s. The process of mitosis was elucidated in 1880s, but Mendel worked with pea plant in 1860s, 20 years before the discovery of chromosomes. So just imagine the intelligence of this person. He is working with genes and the chromosomes which provides a seed to the genes are not yet discovered, and he is working on genes. Now, before Mendel, there were, of course, other geneticists or other scientists who were working with the breeding of plants and animals, but Mendel got so much fame because of his work. So, why Mendel was able to brought an revolutionary change to this field? What was the approach of Mendel? Mendel used the scientific approach to identify two laws of inheritance. So Mendel worked with pea plants, garden pea, in carefully planned experiments. This was another strength of Mendel. He kept a detailed record of every cross, and then on the basis of this data he was able to formulate two laws of inheritance. Mendel grew up on his parents' small farm in a region of Austria that is now part of the Czech Republic. In this agricultural area, Mendel and the other children received agricultural training in school along with their basic education. As an adolescent, Mendel overcame financial hardships and illness to excel in high school and later at the Olmütz Philosophical Institute. In 1843, at the age of 21, Mendel entered an Augustinian monastery, a reasonable choice at the time for someone who valued the life of the mind. So, because of his religious beliefs, because of his fondness with the religion, he entered one of the monasteries. He considered becoming a teacher but failed the necessary examination. In 1851, he left the monastery to pursue 2 years of study in physics and chemistry at the University of Vienna. Now, these were very important years for Mendel's development as a scientist because he was in company of two strong motivational scientists or professors. One was the physicist Christian Doppler, who encouraged his students to learn science through experimentation and trained Mendel to use mathematics to help explain natural phenomena. So, Mendel learned mathematics from Christian Doppler. Another scientist was a botanist, Franz Unger, and Mendel learned the techniques to breed plants and study variations in plants. So the company of a botanist and a mathematician incorporated some skills in Mendel's life as a scientist, and then he later on used these skills to arrive at the two laws of genetics. After attending the university, Mendel returned to the monastery and was assigned to teach at a local school where several other instructors were enthusiastic about scientific research. So, after his studies at the University of Vienna, he returned to the monastery where he also enjoyed the company of individuals who were interested in scientific research. In addition, his fellow monks shared a long-standing fascination with the breeding of plants. Around 1857, Mendel started his breeding experiments with pea plants. Although the question of heredity has long been a focus of curiosity at the monastery, Mendel's fresh approach, his experimental and quantitative approach, allowed him to deduce principles that had remained elusive to others. So, the skills which he learned during his early life, during his stay at the University of Vienna, were employed during his experiments. One reason Mendel probably chose to work with peas is that there are many varieties. So the pea plants have many varieties. They have heritable variations, and that is something which always fascinates geneticists. For example, one variety has a purple flower while another variety has white flowers. Now, what is a character? A character is a heritable feature that varies among individuals. For example, flower color in humans, you can say skin color, hair color, eye color. These are the characters. These are the heritable features which are inherited from one generation to another generation. And Traits are the different variants of a character. For example, the skin color is a character, the brown skin color, the white skin color, these are different traits. Similarly, hair color, white hair color, black, brown, blonde, these are different traits. Another advantage of using pea plant was that it has a very short generation time and it can be grown easily. There are different characters to be studied. It has a short generation time. So these were different characters, properties of the pea plant which make it an ideal model organism for genetics related studies. Furthermore, Mendel could strictly control mating between plants. So this pea plant not only undergo self-fertilization but cross-fertilization can also occur, and furthermore the mating can be controlled. For each pea plant flower has both pollen producing organs, stamen, and an egg bearing organ, carpel. So both self-fertilization can also occur, that is the transfer of pollen grains from stamens to carpel of the same plant or the same flower, and then cross fertilization can also occur. In nature, pea plant usually self-fertilize, that is, pollen grains from the stamen land on the carpel of the same flower, and the sperm released from the pollen grains fertilize eggs present in the carpel. So this is self-fertilization, where the male and female parts are both present on the same plant. To achieve cross-pollination of two plants, Mendel removed the immature stamens of a plant before they produced pollen and then dusted pollen from another plant onto the altered flower. Here you can see removed stamens from purple flower, and then these stamens were transferred to the carpel of the white flower. So this is cross fertilization. His method allowed Mendel to always be sure of the parentage of new seeds because he kept a detailed record for each cross. Therefore, he was able to study the progeny of different traits. Mendel chose to track only those characters that occurred in two distinct alternative forms. For example, tall versus dwarf, yellow versus green, flower position axial versus terminal, seed shape round versus wrinkled. So there were two distinct and alternative forms, and there was no intermediate. He also made sure that he started his experiments with varieties that were true breeding, True breeding is a variety which is homozygous for all the traits under consideration. Which is homozygous means either all the alleles for all the traits are in dominant form or all the alleles for all the traits are in recessive form. So first he produced true breeding varieties, and thereafter he crossed the true breeding varieties with differing traits with one another and observed the F1 generations, and later on self pollinated the F1 offsprings and observed the F2 generation. For example, a plant with purple flowers is true breeding if the seeds produced by self-pollination in successive generations all give rise to plants that also have purple flowers. So a true breeding variety is one which faithfully transfers all the traits to the next generation, or which is homozygous for all the traits under consideration. In a typical breeding experiment, Mendel cross-pollinated two contrasting true breeding pea varieties. Now, first he prepared the true breeding varieties and then he crossed the true breeding varieties differing in one trait or two traits with one another. So the purple flowered plants were crossed with the white flowered plants. You can see here on the right side of your picture. And then when the seeds were produced, the seeds were sown and then the flower color was observed. This mating or crossing of two true breeding varieties is called hybridization. The true breeding parents are referred to as the P generation or the parental generation, and their hybrid offsprings are the F1 generation or the first filial generation. The word filial is from Latin, which means son. Allowing these F1 hybrids to grow and self-pollinate and which alternately produced F2 generation, which is the second filial generation. The Mendel usually followed traits for at least the P, F1 and F2 generations. Had Mendel stopped his experiments with F1 generation, the basic pattern of inheritance would have eluded him. So he followed the crosses till F2 generation, and he didn't concluded his crossing experiments with the F1 generation. And because of this factor, he was also able to study the behavior of the recessive alleles, because the recessive alleles in his case showed their effect only in F2 generation and they were camouflaged in F1 generation. Mendel's quantitative analysis of the F2 plants from thousands of genetic crosses like these allowed him to deduce two fundamental principles of heredity, now called the law of segregation and the law of independent assortment. So after his experiments when he collected the data, when he analyzed the data, he was in a position to formulate his findings into two laws of genetics. Now we study the first law of genetics, which is the law of segregation, also known as the law of dominance. The explanation So now we discuss the first law. Two true breeding varieties, one having purple flowers, the other having white flowers, when they were pollinated with each other, and then the seeds were procured and the seeds were sown, so the plants produced from these seeds, all of them had purple color. Now if there was blended inheritance, then the color of the F1 plants would not have been purple, and it would have been a mix of both the parents' color. But here the phenotype of the dominant parent is being expressed, and the phenotype of the recessive parent is masked. Then these plants were self-pollinated, the seeds were procured and the seeds were grown, and in F2 generation the ratio of purple flowers to white flowers was 3:1, that is 705 plants had purple flowers and 224 plants had white flowers. So when the alleles responsible for determining the white color appeared in homozygous form, then this trait also appeared, whereas here the allele, the dominant allele showed its effect, and it masked the effect of the recessive allele. So what happened to the white flowered plant's genetic contribution to the hybrids? Why there is no effect of the white flowers or the white plant in the F1 hybrid? F1 hybrid is only exhibiting the trait of the dominant parent. If it were lost, then the F2 plants could produce only purple flowered offsprings in F2 generation. So the allele determining the white flower color is there in F1 generation, but because it is a recessive allele, therefore it cannot express itself in heterozygous form. But in F2 generation, when the recessive allele became homozygous, then it was in position to express the phenotype it controlled. Therefore, the white flowers appeared in F2 generation. But when Mendel allowed the F1 plants to self or cross-pollinate and planted their seeds, the white flower trait reappeared in the F2 generation. Mendel used very large sample size and kept accurate records for his results. 705 were purple and 224 were white. So large sample size is always important because if the sample size is small, then you cannot conclude, or your conclusion cannot be 100% accurate. These data fit a ratio of approximately 3 purple to 1 white. In Mendels terminology, purple flower color is dominant because it expressed itself in heterozygous form in F1 generation, whereas white flower color is recessive because it doesn't express itself in heterozygous form. The reappearance of white flowers in F2 generation was only possible when the alleles determining the white flower color became in homozygous form. So there was no dilution. If you recall from the theory of blending inheritance, they say that the two parents' characters are diluted and they give rise to another character in the offspring, which is a mix of both parents. But here you can see that the allele responsible for determining the white flower color is still there in F1 generation and it is only expressed in F2 generation. So the allele does not lose its individuality. Instead, it had been hidden when in the presence of the. So a recessive allele cannot express itself in heterozygous form. It will only express itself in homozygous form. In F1 offsprings, there was a dominant allele which was determining the purple flower color, and this dominant allele masked the effect of the recessive allele. Now these are the different traits which Mendel studied: flower color, purple, white, then we have seed color, seed shape, pot color, pot shape, flower position, stem length, and these are the results of the F2 generation: 705 purple, 224 white, and then you have 6022 yellow, 2000 green, 5474 round, 1850 wrinkled. So there you have this phenotypic ratio. It's genotypic ratio where these are the homozygous, these are the homozygous dominant, heterozygous dominant, and homozygous recessive. Similarly, these three values refers to homozygous dominant, heterozygous dominant, and homozygous recessive. For example, when Mendel crossed a true breeding variety that produced smooth, round pea seeds with one that produced wrinkled seeds, all the F1 hybrids had round seeds because it is a dominant trait. And in the F2 generation, 75% of the seeds were round, whereas 25% were wrinkled, thus ending up in a ratio of 3 to 1. Now let's see how Mendel deduced the law of segregation from his experimental results. In the discussion that follows, we will use modern terms instead of some of the terms used by Mendel. So, because the genes were discovered in 1909 by Johansen, therefore, at the time of Mendel's experiments, he used the word factors to refer to genes. So this is the end of the first part of this lecture. Kindly refer to the second and final part of the lecture in another video. Thank you.